So our next speaker is in person. Joining us now is Dr. Oliver Tobin. Dr. Tobin is an associate professor in the Department of Neurology, Division of Multiple Sclerosis and Autoimmune Neurology at Mayo Clinic in Rochester. His primary interest is MS and related disorders and neurologic complications of histiocytic disorders. He's the director of the Mayo Clinic Rochester Multiple Sclerosis Fellowship and is a former Teacher of the Year award winner in the Department of Neurology. He has active NIH-funded research investigating tumefactive MS and metabolomics and genetic biomarkers of MS progression and activity. Thanks for being here, Dr. Tobin.
Thanks very much, Radhika, and thanks to everyone who's here and everybody who's online. There's some really great talks this morning. Mine will not match that, but I'm going to basically just wanted to talk to you today about the infrastructure available within our Center for Multiple Sclerosis and Autoimmune Neurology and how that could potentially be applied for ADLD research and related research. So the mouse is working. So the center is located in Mayo Clinic in Rochester, but based in all three sites, Mayo Clinic Jacksonville and Mayo Clinic Florida. And the mission of the center is to improve the lives of patients with multiple sclerosis and autoimmune neurological conditions. As part of that, because we're evaluating patients who have essentially the commonest leukodystrophies and other autoimmune neurologic conditions, we see patients who have genetic leukodystrophies as well. And the mission of the center is twofold, essentially to stop diseases that are in progress and then to restore function in patients who have MS and related disorders. And these are two related but separate concepts, as was alluded to earlier, in that if we halt progression of these disorders, patients are still often left with significant disability. And so research and work going into halting or preventing worsening in these diseases is probably not enough, and we need to work on functional improvement as well. And the functional improvement, there's probably a lot of scope to translate functional improvement from commoner diseases into rarer diseases such as ADLD. So our repository, as I say, is based in the Mayo Clinic sites, and the health system is here primarily in Minnesota, Wisconsin, and Iowa. I think there's over 50 sites in the health system, and then we also have Mayo Clinic in Arizona and Mayo Clinic, Florida. The samples are collected from patients both in clinic and remotely. During pandemic, the kind of remote sample collection was significantly increased. And we've over 4,000 patients enrolled with multiple sample types on all patients: DNA, RNA, serum, plasma, PBMCs, and a smaller number with CSF and fibroblasts, and have a pipeline for iPSC production from the fibroblasts as well. So we have 62 patients currently with iPSC lines derived, and over 350 with skin fibroblasts. We also have some whole brains from autopsies, 9 of those, and we have a related study with 770 or so biopsies from patients with multiple sclerosis and related disorders. So almost 150,000 aliquots of biospecimens. These are the diagnoses that are available within the center. So primarily multiple sclerosis, but a significant portion of other disorders as well. And like I said, we have enrolled multiple rarer disorders, and I'll talk a bit about the histiocytic neoplasms later. And then we've more recently have started to focus on serial collection. So what we found initially was that we were collecting single samples on patients, but really we want to see how these patients progress over time, both clinically and from a biomarker point of view. And so we've over 150 patients with serial sample collection, and have a CSF serial sample collection study ongoing for local patients within Rochester as well. These patients have a significant amount of clinical data available. So we have a deep search facility that we can search both the sample biorepository, but also the linked medical records, including full text from all the notes and the MRIs. So patients, almost all patients have OCT and MRI. If you're not familiar with OCT, it's a laser scan, essentially like ultrasound of the eye, looking at the thickness of the different retinal layers. And it's very useful in the context of multiple sclerosis, but also in other white matter disorders, we can identify optic atrophy in an objective manner. So part of the way we give out samples is that we request that researchers give us data back once they have their studies completed. And so that means that we have multiple different data types available. So we have, early on it was haplotyping. Now we have whole exome sequencing, genome-wide genotyping on almost 3,000 patients. We have metabolomics on over 500, a GWAS on almost 200, and then various other data types, including miRNA in serum and plasma, TSPO PET on brain, serum and CSF neurofilament light chain testing, neural antibody testing, cytokine testing, phage display, and O-link proteomics. And almost all of these, with the exception of the miRNA and the metabolomics, are done internally at Mayo in Rochester. In addition, we perform gait analysis, both on single sample patients and on patients with serial collection. And I'm really interested to see the gait analysis talk, which is going to be next. So the platform that we're using is an APDM. It's inertial sensors that are placed here. You can see them on one of our researchers here, so on the chest and on the arms and legs. Patients walk up and down in the corridor. And on the right-hand side here, we have a 3D video analysis. I get it working. There we go. What you can't get with the inertial sensors is limb angles. And so the 3D video gives you limb angles as well. So that's what that's for. So this is pretty quick to do. So you can do it in about 20 minutes. It's done in the clinic. And so when we see patients clinically, the research coordinators come up, enroll them into the study, and do the gait analysis immediately when they're there. And so every time the patient comes back, then we do sequential analysis, or at least data acquisition. And so this is our coordinator team. So we've got a large group of coordinators in Rochester, which is fantastic because they will come up essentially immediately when we see a patient, we call them. And then we've one in Jacksonville and one in Scottsdale. And in addition, then we have our colleagues from the Center for Regenerative Biotherapeutics who perform our skin biopsies for fibroblast collection. And there's biostatistics support, which is available through the center as well. So if people want to use samples, you can do that, but you can also use samples and data and have statistical support through the center. Another exciting collaboration that we have is with our AI group in the Department of Neurology. And so one of the things that we noticed in some of our histocytic patients was that they have progressive dysarthria. And it was very difficult to quantify this over time. And so this is a way of doing that. So it's essentially a remote speech. So the patients can have a speech sample that's captured on their phone. So it can be done at any time, any place. It's uploaded to the Mayo Clinic server, and then it has both a automatic analysis and a human analysis done by speech pathologists. So we have speech pathologists who code the different parameters of the speech, and then the AI machine gives quantitative parameters as well. And so the overall goal of that study, which has been run by Dr. Hugo Botha, is to enroll 100,000 patients, 100,000 speech samples from 100,000 patients. And so we've collaborated with his group to collect serum, CSF, gait analysis, and speech analysis on our patients within the MS Center as well. So we've developed an internal tool for searching. What we found initially was that we had several thousand samples and couldn't find them and didn't know where they were. So we had to develop a tool for searching. So we've linked the studies that the patients are enrolled in, what samples they have. That's linked to the clinical images, both the MRIs and OCT studies, demographics, that's kind of easily searchable by our coordinators. So if any either internal or external collaborator wants to kind of see how many patients we have with certain parameters, they can kind of search that and have an answer within a few minutes. So that's just an example of how they do that. And then what's really nice then is the deep searching that's available through Mayo Data Explorer. So the Mayo Data Explorer is a search tool for the Mayo Electronic Health Record, and that searches all sites. So the Mayo Clinic Health System sites within Wisconsin and Minnesota, Iowa, and so on, and the Rochester, Scottsdale, and Florida. And so, for instance, if you search for multiple sclerosis, there's about 20 to 30,000 patients with MS who've ever been seen. And so you can search all the parameters on them. You can search basically anywhere there's text. You can do free text search, and they've integrated large language model analysis into that as well. So we've multiple different collaborators. There's just a selection of them here. For our academic collaborators, as I say, we don't charge for the samples, and if there's a reasonable request, they're generally approved. We generally require that some reasonable data is submitted back to the center to improve the quality of the data that's here, sometimes, but not always, some sort of academic involvement in the study, depending on exactly what the study is. So just talking about rare diseases. So this is one of my interests is in histiocytic neoplasms. These are rare, supposedly rare, myeloid lineage neoplasms that cause persistently enhancing brain lesions. And so these patients are often diagnosed with neuroinflammatory syndromes because the biopsies, particularly brain biopsies, look inflammatory. But it was shown, yes, about 2 decades ago that the myeloid clones or the myeloid cells that are in these inflammatory infiltrates actually have mutations in the BRAF-MEK-ERK pathway. And so these were reclassified as neoplasms, and they actually respond beautifully to typically to MEK inhibition or BRAF inhibition, depending on the underlying somatic mutation. So this is a cohort of patients that we wanted to evaluate. And so we've enrolled all of these patients into the Center for Multiple Sclerosis and Autoimmune Neurology Biobank, which allows us to do gait analysis on all these patients, speech analysis, to serially collect their MRIs and to serially collect blood and CSF on these patients. So this is just a recent study that one of my fellows did looking at serum neurofilament light chain in patients with histiocytic neoplasms, showing that it's elevated in patients who have parenchymal enhancing lesions more so than meningeal or dural-based lesions, and showing that the extent of serum neurofilament light chain correlates with the extent of enhancement over time. So showing that that's potentially a good marker for response to treatment in histiocytic neoplasms. As I said, we have a particular interest in the restore aspect of the center's mission as well, and stopping the disease is probably not enough. And so we've collaborated with our Assistive and Restorative Technologies Lab, Dr. Kristen Zhao, here based in Generos, actually in St. Mary's campus. And this is a really large, beautiful lab that she has over there, where we're essentially implanting spinal cord stimulators in patients with multiple sclerosis to try and improve their gait function. And so this is a bilateral epidural lead placement occurring in our first patient enrolled into the study. These patients have stimulation done. They're essentially, the technology is essentially like pain stimulators, but the buttons that you fiddle with on the device are different. Right? The stimulation parameters are different. So this is an example of a man who has stimulus-induced clonus in his leg, and the stimulation was off on the last iteration and is on now. And you can see that the clonus is completely removed with spinal cord stimulation. So it essentially goes back to normal after the stimulation is turned on. We've also looking at gait function. These patients. Let's get this running. So we've got no stimulation on the left-hand side, stimulation on the right-hand side. And if you look at the stiff leg, which is the right leg is essentially the spastic leg, with stimulation, you see the knee angle bends more. We get improved gait function with stimulation. That's with stimulation and 4 weeks of rehabilitation. And so we can show that even if you turn the stimulation off after the 4 weeks of stimulation and then turn it back on again, the effect goes away when you turn the stimulation off, and when you turn it back on, the knee function improves. So our goal is to do longer rehabilitation with longer periods of spinal cord stimulation, essentially, for those patients. So again, that's something that it would be very difficult to do that for rare patients, like patients with ADLD, but as we've heard earlier, these patients with ADLD, they definitely have significant core dysfunction, and so spinal cord stimulation for improvement of gait function may well be a good therapy for these. The other question then is for rare diseases. How do we deploy trials that patients are rare, and how are we going to get patients to travel to Mayo Clinic? And so the Department of Neurology has set up a collaboration with the NeuroNext collaboration, which is a multi-center collaboration for clinical trials for rare diseases. This is coordinated through MGH with a data coordinating center at the University of Iowa. And this Extinguish trial is the first trial that we've been involved with through this mechanism. And this is a trial of inebilizumab, which is a CD19 inhibitor for NMDA receptor encephalitis. So again, a very rare disease. This particular disease, if you don't treat it early, it's probably going to have a poor outcome. So getting patients to travel long distances is probably impractical. And so this multi-center trial network has been helpful to set that up. So as I said, we generally approve any reasonable requests for sample or data with fairly little fuss. If you're interested in requesting data or sample for internally, request from the CMSAN website. There's a really hard to find hyperlink on it there, but it is there, right there. Anyway, or just email me or one of the coordinators. There's a brief form to upload, just outlining the request, and then it's reviewed by our group of Biospecimens Access Committee. We've tried to streamline this process, so only 3 out of the 5 people need to review that now, and so we generally get the requests reviewed in about a week. So if you do have a request, we should get a response very quickly. And then, as I mentioned, we have biostatistics support services as well, and funding for that, so that if you require that as part of your study, we can offer that too. So as regards control samples, we're collecting control samples ourselves as part of the center. This wasn't our focus initially, but we realized we need controls now, and we've got controlled CSF and serum from patients with NPH, migraine, and functional disorders. And then several of these are other projects then are essentially within SIM. So the Mayo Clinic Biobank has 50,000 patients, again, with deep clinical data available. The Tapestry study is within SIM. It's 10,000 Mayo Clinic patients with whole exome sequencing. These are patients who have a longitudinal Mayo Clinic follow-up. And then Project Generation, the Regeneron project, is 100,000 patients with whole exome sequencing. And these are patients who come from clinical studies. So whereas Tapestry, the patients are essentially enrolled clinically. For Regeneron, they're like, we've sent essentially 2,900 samples from the CMSAN to the Project Generation study to get whole exome sequencing, and we'll be able to compare that with the other 100,000 that are available. There's also the Sangre Pour Salud there's a link with them, a Latino population in the Southwest US, and Biobank, Mississippi, which is important for improving our diversity. So that's all. I'd be happy to take any questions. Thanks very much for your attention.
In the slide where you showed the diagnosis that you had a thing on leukodystrophy. Were these patients included as being autoimmune disorders and kind of given the diagnosis of a leukodystrophy later, or they were included when they had a diagnosis of leukodystrophy?
So generally what we try and do is enroll people right when they come. It's the best time to enroll someone is when they're there with you in the clinic. So most of the time you don't have a diagnosis in that setting. So if you have somebody, so we focus on longitudinal assessments of patients with progressive MS, because we want to focus on basically curing progressive MS, and then patients who are undiagnosed, so patients who have unusual syndromes. And so for those patients, we don't necessarily know what they have when they come in the door, but that's the time to enroll them because that's the time that they're there and you're going to get their blood draw and everything. Most of them, as you know, don't come with a genetic leukodystrophy diagnosis. We're usually working on it for a long time to get it.
So do you think that you could have some?
Yeah, so I mean, we're going to get the Regeneron data next week or the week after. That's what they tell me. Yeah. So we'll have the guts of 3,000 patients there, and I think that's where we need to look to see do we have any other genetic leukodystrophies within not just genetic leukodystrophies but autoinflammatory syndromes as well. I mean, we've only very recently been widely using autoinflammatory panels, and even there, the in vitro autoinflammatory panel didn't include presenilin. We're seeing a reasonable number of patients with amyloid-associated angioitis. That's everything. So I think there's a lot of scope to look closely at this cohort.
And the gait analysis is done same way for any diagnosis.
Yeah. So that's great. So we have it's the same gait analysis. So the parameters that we're looking at the moment are mostly sway, gait speed. You can look at asymmetry. I think there's a lot of scope. If you think about the ADLD population, there's a lot of scope to do kind of home-based assessment. So a quantitative actigraphy, so essentially like step count. How active is somebody over time? And does that change with response to treatment? But what we're doing in clinic is essentially the inertial sensors. We get them to stand, to walk, assess their stability and their asymmetry. And we have both an engineer from Kristen Zhou's lab and one of our multiple sclerosis fellows are working on that data at the moment, primarily in the MS population right now. But there's a huge scope. There's similar to genetics, there's far more data than there is manpower to do it.
Thank you for the great talk.